So I'm going to turn over to the topic of today's special presentation. So we have a very productive partnership with the Gairdner Foundation. I'm very grateful for that partnership. So every year, RCIScience has the privilege of partnering with one of Canada's premier scientific organization, the Canadian Gairdner Foundation. In addition to recognizing excellence in medical research, the Canada Gairdner Foundation also works to raise the profile of that research among the Canadian public. So again, thank you very much for coming today. So I'd like to turn the podium over to a good friend and colleague, President of the Gairdner Foundation, Dr. Janet Ressant, to say a few words. Janet?
Thank you, Reinhardt, and again, I'm so glad to see everybody here. I am the president of the Gairdner Foundation. I have a really fun job. The Gairdner Foundation has been giving awards for research in medicine and biomedicine that has impact on human health for over 60 years. The Gairdner Foundation is founded in Toronto by a philanthropist, Jim Gairdner, who wanted to recognize research that's had impact on human health. And importantly, he wanted to recognize it internationally, wherever it occurred. So the Canada Gairdner International Awards are given annually to researchers around the world whose research is really at the cutting edge and is having impact on human health. We've given over 380 international awards over the last 60 years. 89 of those have gone on to receive the Nobel Prize, including this afternoon's speaker. So Peter Ratcliffe received the Gairdner Award in 2010, along with his colleagues Gregg Semenza and William Kaelin, for the work that he's going to tell you a little bit about this afternoon. Their work on how our cells sense oxygen. 2010 and last year now, I guess, the same threesome received the Nobel Prize in medicine. So Peter also has been and continues to sit on our medical advisory board. So the reason he's here this weekend is that starting tonight and tomorrow all day, we have gathered about 20 outstanding scientists from around the world to Toronto. We always do it in January. We always pick the coldest part of the year, and we put them in a hotel room, and we discuss all the nominations that we have received over the last year for this year's international awards. So Peter sits on that advisory board, and that's why he's here today, and we're very grateful that, given, I'm sure, what must be a very busy year, having received the Nobel Prize, that he's taken the time to stick with Gairdner and also be here to give this public lecture this afternoon. It is part of the Gairdner mission to really bring science to the public and also to inspire the next generation of students to take up careers in science. So this is one part of the partnerships that we have that we really value, and I'm very happy to be here as a member of RCIS myself to promote this afternoon's lecture. And I'm going to turn it over now to Ron Perlman, who's an advisor for the Gairdner Foundation and a long-time leader in RCIS, to do the formal introduction for Sir Peter Ratcliffe.
Well, thank you, and it's a pleasure, as we've all said, that everybody is here today and has braved the cold and the snow to come to this exciting afternoon public lecture. Sir Peter Ratcliffe is a physician and scientist who's known for his research in the regulation of erythropoietin, a hormone that stimulates red blood cell production in response to low blood oxygen levels, and for his research into the mechanisms cells use to sense oxygen. You've heard a great deal about prizes and things. I'll just mention that his undergraduate training was at Cambridge University, and then he studied medicine at Cambridge and St. Bartholomew's Hospital, and he trained in renal medicine at Oxford, initially working on the physiology of renal oxygenation and its implications for renal injury in shock. He has many awards. You've heard about some of the most prominent, which I'll mention briefly. Janet has already mentioned. In 2016, he took up the position of Director of Clinical Research at the Francis Crick Institute in London, and in June of that year, he took up the position of Director of the Target Discovery Institute at the University of Oxford. So as you've heard, he, along with Gregg Semenza and William Kaelin, were Canada Gairdner International Award recipients. And this past year, the three of them were also awarded the Nobel Prize in Physiology or Medicine, and the citation for that award was for their discoveries of how cells sense and adapt to oxygen availability. So it's a pleasure, as Janet has told you, the reason for Sir Peter being here, that we are able to have him present this public lecture to you this afternoon. Peter.
So thank you for that very kind introduction. This will be a mixture of personal anecdote and science that I hope will give something to most of you. The work pertains to oxygen homeostasis and our findings of certain mechanisms by which cells signal and transduce that signal to affect oxygen homeostasis. The subject started, or at least the phenomenon of adaptive responses to low oxygen, started with the environmental physiologists of the late 19th century, early 20th century. And that's where I'd like to begin. John Scott Haldane led this expedition to Pikes Peak, Colorado, to understand altitude adaptation. But the heroine for this lecture is surely Mable Fitzgerald, the only female member of the party, not allowed to be a member of Oxford University on grounds of her gender, but seen here with the others at the summit of Pikes Peak, where there was a rather comfortable hotel which enabled the party to make its laboratory setup. However, it was decided that Fitzgerald would work elsewhere on her own, not at Pikes Peak, but at the mining towns of intermediate altitude. The story is this is because the men were concerned that she was unchaperoned in that comfortable hotel, so they sent her away on her own to the mining towns of what was recently the Wild West. Oxford eventually recognised her with an honorary degree at the age of about 100, I believe. So she was quite a tough cookie. And these are the measurements that she made in those mining towns of the change in hemoglobin and the change in carbon dioxide, reflecting the respiratory rate at different altitudes. And they record the extraordinary sensitivity of both of those changes to even modest changes in altitude, and hence by implication oxygen. This wasn't the first evidence of adaptation to low oxygen, but it was, I think, the first evidence of the extraordinary sensitivity of this response, which drew us to the field. Now, during the 20th century, the work of Alan Ersklev and others established that this response was made, at least in the blood, by production of the hormone erythropoietin, which regulates red cell production. And that, too, is extraordinarily sensitive. Here you can see the response to just donation of a unit of blood, so hardly a major stress. And it was, the EPO is made in the kidney. I am, as you've heard, a kidney specialist by training, and it was that extraordinary sensitivity to hypoxic challenge that drew me to the field. There was a new opportunity, and that's always important in science. The hormone, the gene encoding EPO had been cloned and was available for study. And we decided that we would try to move backwards from the gene to what we thought would be a very special sensor in very special cells within the kidney. And that's as I'm a kidney specialist, that's where we attempted to start. The problem was that those cells were not available in a tissue culture dish, and that's what you need for doing the studies that I'm going to describe. So we tried to provoke them to grow by a trick which is to express an oncogene, a cancer-promoting gene, inside the EPO gene, and we believed that this would make a tumor of the EPO-producing cells, and this would enable them to grow in a culture dish, and then we could study them and study, we thought, their very special properties as oxygen sensors. The experiment failed in that they still would not grow in that tissue culture dish, but it did identify them as the kidney fibroblasts. The kidney, I'm a kidney specialist, so you'll know this. The kidney has all sorts of really interesting cells in it. They do the filtration, they do the reabsorption of the filtrate, so on. Every interesting cell in the kidney had been said to be the producing cells, but except for these, the lowly and slightly boring fibroblast. But you know, that should have been a clue. But it wasn't in the end, nor did we get the cells. So credit for opening this field should actually go to Franklin Bunn in Harvard, who did often the way in science, the most simple things matter in changing the field. It was a central problem to get that tissue culture model. That is central in science. You must have the right system for the technology to work. Frank took some hepatoma from a liver cancer cells, and what he found was that if you made the oxygen lower in tissue culture, these cells would then make EPO. EPO is made by the liver and kidney, so we thought the special sensors were in both the liver and the kidney. The extraordinary thing about this, most of you know physiology, will know you'll die well before you get down to these concentrations. This is Mount Everest here, so you know that's about as far as you can go. So it was unusually low, unexpectedly low, but the tissue culture is different from the way the atmosphere you breathe, so there are ways to understand that. But the point of the story is you institute something a little bit unexpected, but that simple observation opened the field. And you've heard of Gregg Semenza, another physician scientist, Jaime Caro, said about using Frank's cells to find the sort of address signals at the EPO locus that would make it respond to oxygen. We wanted to find the DNA that codes a gene, but then you have these regulatory sequences. And we worked on the mouse gene, cutting and pasting, chopping it up, and finding which sequences would confer oxygen regulation on another protein. We knew that once we got there, that was the first stage in the journey. They must interact with something that was lying upstream relevant to the sensing of oxygen. And what we found was this sequence lying just at one end of the gene could confer that response. This is an assay of gene expression when the cells were made, when Frank's cells were made low in oxygen. That was a lot of work, very laborious, but it was just the first step in the journey. Those cells must be interacting with something. So what was it? I'm a rather impatient person by nature. So I also wanted to take a shortcut. I wanted to do something which I thought would be very clever and expression clone the upstream thing. Now, some of you are scientists, some of you less. I'll explain expression cloning. That means that we're going to take genes from a cell that has the oxygen sensing property and transfer them into one that doesn't. And the genes that transfer that property are going to be the ones that we want. And it's, you can do these very clever systems. We all like to be clever. and we all like to take shortcuts, and that was me. This is what we wanted to do. So we thought that Frank cells were the oxygen sensing one. We'd transfer the genes to an oxygen insensitive cell. That would be practically every other cell in the body, because this is this highly specialist thing. You have to do control experiments, so I'll look very carefully to make sure that the oxygen insensitive really was insensitive, and it wasn't. It wasn't. They completely destroyed the strategy, but today I can remember that radiograph. You know, the moment you remember coming out of the developer in the John Radcliffe Hospital. Goodness me. A few minutes later, then it was initially irritating, but we realized that there could be something quite important, and you might think what that might be. But we could go on and actually show that all range of other cells have pretty well the identical property they're made from the skin, the lung. Things got nothing to do with making EPO. So rather than this being a really specialized thing, must be everywhere. The implication is, I talked about Fitzgerald's very sensitive work, there must be lots of other things beside control of the blood that was, again, conducted with that extraordinary sensitivity. And the implications for that, for understanding how the body works, were rather profound. And we got very, very excited about this. And this is the experiment that changed my life, and the reason why I'm here for the Gairdner Foundation, and so on and so forth. Not being one to take chances, we wrote this work up, and I had a rather nice sports car as a young kidney doctor, and I drove to the offices in London of a very famous journal in Little Essex Street, and Maunday Thursday, day before Easter, 1992, and I imagined that I'd be able to burst into the office and see an editor, and that editor would, like myself, immediately understand the importance of the work and publish it, no doubt, after Easter. This is the result. This is the letter we got back for them for about 4 months later. Of course, many people have had these rejection letters, and they store them up for when they get the famous call to Stockholm. For the young people, this is the important bit. Look what they wrote here. It took them 4 months. We had great difficulty obtaining referees' reports. Now, for the young people in the audience, that's an important thing. It means you're in a field of your own, and this is going to make life a lot easier for you, and that if I have one message to the young people, you must pick your own subject, and of course you'll have these rejections you'll have to wear with them. We put the letter away, and obviously there was a reason for that. We knew it was important, and we thought we might get that letter out again, and so we have. So that was it. I believe this was important because it implied the same system was being used in all sorts of other cells for other purposes. My colleague and co-recipient, Gregg Semenza, did the beautiful work of finding the next bit. That was the molecule, the protein that stuck to that control sequence that he and we had identified. He called it HIF, hypoxia-inducible factor. So the question now was, what other genes did HIF regulate? And we found the first one of those. This was phosphoglycerate kinase. That's a metabolic enzyme. Here you see Gregg's HIF. This is binding EPO, this bit here, and you can see the same bit binding phosphoglycerate kinase controlled sequences. So that was a little bit of a surprise because it connected this very dynamic EPO response to something we thought was quite stable, metabolism, but the assumption was it probably wasn't, and that too was being tuned by hypoxia. Now these primitive animals don't make EPO, they don't even have a blood vascular system, but this type of metabolism is prevalent in pretty well all forms of life. So we thought this might be a very ancient system, and then that's the next step, looking primitive fruit flies and worms. We'll come to fruit flies and worms and why you should know they're very important later on, because they were the solution to the problem. So this was in the fruit fly cell, and again, we saw the same thing. We saw HIF in these cells made from insects, from flies. Of course, you might not see that as very important, and I have to say, nor did the reviewers of that paper. It had the distinction of being first lost and then rejected by an editor of the Journal of Biological Chemistry, normally quite a good journal, but they were not so efficient on that occasion. So that meant that it was in all cells, all animal cells, irrespective of whether they make EPO, and it did all range of other things. And in the intervening 20 years or so, we're now aware that HIF regulates a whole range, hundreds of thousands of responses that the body might work to control its oxygen supply and needs and responses. I'm not going to talk about that for the moment, because the problem at the time was to move upwards from HIF now to the next bit, and that's the same sort of strategy. Take a bit of HIF, link it to a protein that didn't have oxygen-sensitive properties, find the bit that conferred oxygen sensitivity. That bit must be the next step going to the oxygen sensor. So that's what we did, and we found these 3 bits in HIF all could confer oxygen sensitivity. And remarkably, this had been known for many years, that cobalt poisoning sometimes occurs in beer manufacturing. They use cobalt to change the froth in beer, poison the beer drinkers from time to time. They get a high level of blood, rather remarkable. So cobalt had something to do with this, and all these bits would confer response to cobalt as well as oxygen. So we knew that they were the bit that connected with the oxygen sensor. Now, everyone knew that a thing called protein phosphorylation, putting phosphate groups on proteins, taking them off, was a signaling system. So we mutated, changed every amino acid, every residue in this bit to take out all the phosphoacceptors. It made not a blind bit of difference to the way it worked. Again, you can imagine the reception of that with our editorial friends. You know, this is all negative data. Someone actually chanced to say that they thought the lab was overfunded. So why had we gone to the trouble of making all these mutations when none of them worked? You know, that's okay. But it did exclude the main hypothesis. And that is important. Exclusion is important in science as well as proving things. So what was it? Now, this was the next step. We made the connection between that bit of HIF and a protein called von Hippel-Lindau, which Bill Kaelin, the other co-nominee, was working on. And this is one of those eureka moments, you see. Cells that had this VHL protein, they regulate HIF with oxygen. Huge difference there. And when they're knocked out, they don't do it. So VHL is absolutely required for this, and it's touching that sequence. So that was a eureka moment, but it didn't quite start like that. Bill, you had been working on this before. Some of the interviewers for this Nobel thing said, did you all work together in the same lab? Or did you ever compete? And I thought, that really threw me. We're amongst the most competitive people on the planet, like most other scientists. So I'll just talk you through this. It's a sort of competitive symbiosis, how scientists work. So this slightly more complicated story. See if you could stay with this. So Gregg and I had identified multiple genes that were induced by low oxygen. That was available, that information, Bill and Rick Klauster and others worked on those. They found that some of them were upregulated in cells that were deficient in VHL, as you would anticipate. But they didn't understand the mechanism. Despite the greatness of Harvard University, they could not get the mechanism. And you know, this is one for the UK. So what as recognised by the Gairdner Foundation, why was that? We heard this news at a meeting. We rushed back to blot for HIF. HIF's the obvious candidate. This is what we saw. Nothing at all. And I assume that's what they saw in Harvard, which is why they couldn't go forward. Another line, Steve McKnight was looking for things in the human genome that looked like HIF, so-called paralogs. He found one, but he didn't really show it was a paralog because HIF's regulated through those changes in protein that you saw. Need an antibody to measure that, and for the deans and also for the students. I think you should have an antibody making system before you're allowed to run a research institute. And we did. And the reason is, if you want to lead a field, you must make your own antibodies. By the time you wait to buy them, which is expensive, other people will catch you up. So we had made the first antibodies against not HIF1, but Steve's paralog, HIF2. It was the HIF2 that gave the game away, because many of those cells, for reasons I can't explain, can't explain to you today, it would take a little time, only express HIF2 through them, and we made the key connection there. Must have been very irritating for them. So there are all sorts of implications. You know, as I said, why did this kidney cancer switch to HIF2 as a fundamental lead in the way cancer works? That HIF2 is now being used to make treatment for kidney cancer. I'm not going to have time to do that. What we want to do here is go up the oxygen sensing pathway. And this was the next connection. What would it be? So this is where we now know that this HIF thing that Gregg identified sticks together with the VHL thing. That's a protein destroyer. So in the presence of oxygen, the two things stick together and they're destroyed. In the absence of oxygen, that doesn't happen. The HIF builds up and it produces all those responses I talked about. So the question was what was regulating that? And with the classic biochemistry issue, we knew that treatment of the HIF with an extract would make that happen. So there's something in that extract. I'm not a biochemist, and this is where the own subject comes. We were able to muddle through this and still have a leading position alongside Bill. A decent biochemist would have done this in short order, but they were working on other questions, fortunately for us. So we took our time and muddled through this. And these are the things we found. It was heat labile, that's a classic thing. Needed oxygen, that was quite difficult, have to exclude oxygen from the buffers. And so on and so forth. We knew it needed iron, didn't need ATP, consistent with it not being that protein phosphorylation. We were always suspicious of this proline residue. This is an assay for the interaction, and you can see that if that one's mutated, it just doesn't occur. And then this was one of those eureka moments you live for. We synthesized a peptide which had the hydroxyproline in it rather than proline. That one didn't need treatment with the extract. So that's you know, that doesn't happen that frequently. So the result like that, and that again was I can remember the call to me at home on a Sunday morning explaining the result. That's very unusual. In the UK, it means the experiment was done on a Saturday. That's a bit unusual. It means that the result was anticipated. There was an impatience about the results. Someone went in to read it on Sunday morning. That's unusual. What's really unusual is the result was positive. Nearly everything in science is negative. The postdocs come and give you the dreadful news on Monday that it all hasn't worked, but that's a Sunday morning experiment. So why was it important? Because prolyl hydroxylases, that was the implication that the enzyme biological catalyst was doing this. There are other forms, other types of prolyl hydroxylase were known to split dioxygen and use that in the reaction to take hydroxyproline out of proline. And actually, had we read the biochemistry books, we might have been better informed, because they were classically inhibited by cobalt. That was written up in the 1960s, which we ignored, as did everyone else. So the question is, what's the prolyl hydroxylase? Now, the known enzymes modify collagen. That's a structural protein in your bones. It didn't seem credible that that would be the oxygen sensor, and the sequence was not like collagen anyway. So, but people had a prejudice. Well, those are the prolyl hydroxylases. They don't seem really credible oxygen sensors to me, and that stymied us for a while. But you imagined we were looking for another type of prolyl hydroxylase. So I'll just take you through this. It's important for people not in science to know that knowledge builds on knowledge in unexpected ways. So you shouldn't have any doubt about the funding of knowledge creation whose utility is not immediately apparent. And I'll see if I can explain that to you. So we were fortunate in meeting up with this guy, Christopher Schofield. He's been a terrific colleague ever since. Chris was working on antibiotic synthesis, a completely different subject. He crystallized the enzyme that does this, and it had, this is a schematic, it had a particular signature, the structure of that enzyme. Turned out that those enzymes were then related to the collagen enzymes in the way they worked. So using this structure, Chris was able to predict in the human genome a dozen or so enzymes like this, some of which might be collagen prolyl hydroxylases. It's a completely different direction. So how would we ever know that? At the same time, Bob Horowitz, who also won the Nobel Prize, was working on a nematode worm, C. elegans, and they were looking at mutant worms that had a phenotype, an abnormality. In this case, they had difficulty laying eggs. And what this paper describes is 149 mutant worms that had difficulty laying eggs. So where is the story going? Those egg-laying worms were beautifully catalogued by Horowitz, and it's an extraordinarily well-organized community. You don't need an animal license to work on them. Nobody cares about the pain suffered by worms, should they do it. And you can buy them through the post without any experience. You just order them, and they come. So Chris's suggestion included some genes which were in those mutants. But of course, we had to have an assay means, and we had a postdoc in the lab who was a bit restless. He didn't like wet bench work, so I set him off to find the C. elegans HIF. Obviously, and remember the bit about antibodies? We need to detect it. So we made an antibody. And this is what happens with the worm when you make it hypoxic to its HIF. It goes up, rather striking signal. So we didn't need a license. We didn't need any particularly sophisticated equipment anyway. We just used a bell jar to make them hypoxic. And Andy Epstein, who did this work, we looked at all sorts of mutant worms. There's nothing wrong with the oxygen sensing system, but one of Chris's mutants, EGL9, egg-laying mutant 9 in Horowitz's catalogue, was like this. It's completely lost the oxygen sensitivity. Since Chris predicted this was a prolyl hydroxylase, that's what it was, and that was essentially the solution to the problem. So it's rather simple. Your body has this switch, a regulatory prolyl hydroxylase, splits dioxygen, it puts one oxygen atom into proline. Oxygen, as the chemists will know, can hydrogen bond. That hydrogen bonding to the VHL protein promotes destruction. So simple switch. Basically, that's how you work in relation to your blood vessels that make your heart work, your circulation work, your red blood cells, your metabolism, all sorts of things. And that's what the Gairdner and the Nobel Committee recognized. There's one thing good about winning the Nobel Prize. It may be true also of the Gairdner. They never take it away. So once you've got it, then you can argue you didn't really solve the problem after all. There's all sorts of other interesting things still to do. And so the question is, what's more in oxygen homeostasis that we haven't done? And we have described this response, which does hundreds of thousands of things, but it can't do everything that's necessary for the body to control oxygen. That must happen on very short timescales as well as long timescales. We've only got a longer timescale. So what else? This was another Nobel laureate, Cornel Heymans, who described the carotid body, an organ in the neck that controls respiration. And we looked at that organ, and you see this brown staining, that's HIF. So for whatever reason, although this responds far too rapidly for it to be responding to changes in HIF, it contains an awful lot of it. We go back and look at Fitzgerald's work. We talked about the hemoglobin, but she also did the controlling of breathing, that as you adapt to altitude, you breathe more rapidly. But that's a slow response, so you become more sensitive as part of that adaptation. So what about that response? What was that HIF? This is a mouse experiment. We did a surrogate of Fitzgerald's work. We adapted mice to 7 days at about 5,000 meters, and this is the sensitivity of breathing to oxygen goes up about 3 or 4 times. That's what happens if you knock out HIF, though that exaggerated response completely ablated. That this is a drug treatment. These are genetic experiments. So we think that there'll be a reflection. This is ongoing work. Some of you will be scientists and want to know what we're doing at the moment, and that's what we're doing. What is the relation between this system and really rapid responses that can't occur through gene transcription and the conventional way that HIF works? I want to finish up. One other consequence. We say knowledge builds on knowledge. Well, this system is only present in animal cells that we've described, but since then, possibly prompted by our work, other people working in other kingdoms of life have demonstrated that enzymatic protein oxidation coupled to protostasis signals oxygen levels. Seems a rather inefficient way to do things. But the question was, do any of these operate also in animal cells? Are there more things to find out? This is the rather beautiful work largely of Francesco Lacozzi and plants. They're different. They make oxygen. They can't move around, so they have a different challenge. Their challenge now comes from flooding. So if you overwater your partner overwaters your favorite plant and kills it, it's killing the plant from hypoxia. That's what it's doing. It forces the oxygen out, and that's quite a big environmental problem now. The plants use a different type of oxidation to a different amino acid, cysteine, and that also signals for degradation in the presence of oxygen. So I met Francesco, chance meeting in Rome. This is how science works. We just the slightest thing. He said, I'd like to see how your animal oxygen sensors work in my plants. So you can have the bits to do that. Why don't you give me the bits from plants, see how they work in animal cells? And that's what we did. This is a plant sequence, AT for Arabidopsis, a model plant organism. We connect that to a fluorescent protein, put it in animal cells, and see how it goes. So, oddly enough, we found that it was oxygen sensitive in animal cells without even transferring the plant enzyme, that plant cysteine oxidase. So there must be something in animal cells. Moreover, when we mutated the cysteine residue, which becomes N-terminal, it's a bit technical, there's an enzyme that makes it N-terminal, you see it just completely flattens the response. It's different from the HIF response, you see. So there must be a plant enzyme doing that. This experiment I love. You heard from Ron, I'm the clinical director of Francis Crick. So that means I'm supposed to do medical experiments. But this is a plant experiment. This is Francesco's plants. And when he's knocked out those enzymes, they're rather poorly. When we put the human enzyme back into the plant, the little plant grows beautifully. Is that not medically relevant? Well, you know, this is another enzyme. They're classic drug targets, just might be so. These enzymes seem to work on G proteins. This is a little bit technical. G proteins are proteins that are involved in signaling. They're the target of half the drugs in the pharmacopoeia. And this this enzyme that's relevant to the plants regulates the G protein signaling. And here we can see that signaling, that the cells, we can knock out that enzyme. They have upregulation of this regulator, therefore downregulation of the G protein signals, and this is a response to carbachol. That's a famous type of neuromuscular drug. So, in summary, I showed you an animal human oxygen sensor. It does hundreds of thousands of things in the human cell. I showed you a plant oxygen sensor, does hundreds of thousands of things in the plant cell. I showed you that one also has an ortholog in animal cells, working parallel, doing all sorts of things through the G proteins. What I didn't show you is that there's a connection. So as well as being regulated by this system, those G protein signals also regulated by HIF. So this is Darwinian biology for it. It's just pasting bits on, and you're not actually an efficient way of doing what you do. You are fit for purpose, but you're extraordinarily inefficient, baroque way of doing what you do. Everyone knows that wheels on a bicycle make you go faster than running, but fast-running animals use legs because they're trapped by Darwinian evolution to use legs. And you're just baroque creation of bits cobbled together. So what's the point of this? Well, against all this, how could you ever make a drug to treat anemia? How could you ever get one bit out of that complexity and make a drug that would have a medical purpose? Couldn't do it for sure. But what have we got? Well, it seems that with a sufficiently low dose of an inhibitor that blocks up that enzyme, makes the body think it's hypoxic when it's not, we can treat the anemia that's associated with low EPO, that occurs in kidney disease. I told you EPO is made by the kidney, fails in kidney disease. You get these lovely rises in hemoglobin that may take over at least part of the EPO market. That's quite significant. The EPO market's 10 billion a year, so quite small beginnings can lead to quite substantial outcomes. But if you'd asked the field, could this happen at the outset, every informed opinion would say, no, you can't do this. So the moral of drug discovery is this, if you can't always get what you want, but if you try, sometimes you get what you need. Who wrote that? The famous words, Sir Michael Jagger and Mr. Keith Richards, of course by repute they knew a bit about drug discovery. But what they're saying is, actually I have to have a little rail against precision medicine. Molecular science can take you so far. It can give you a target, and that's what we've discovered. The plant enzyme's another target. But then there's an element of empiricism which you can't really close. And of course I could give you all sorts of reasons post hoc why we might be able to, why the kidney's special circulation could make it sensitive, why drugs are concentrated in the liver and kidney, why you might just integrate the response of the red cell to EPO. The red cell lasts 120 days, so if you just get a little burst on the red cell, you might integrate that. But it's all post hoc. I can be very clever and make those reasons up. Truth is, it has to be empiric testing, and failure to appreciate that in the drug discovery industry, I think, costing them billions. Molecular science can and will, and I hope I've just shown you, take you towards a drug. Cannot take you all the way. Drugs rely on dose to get that effect. I'll stop there. Quite a lot of people to acknowledge in the group over the years. This is the current group on the study day, and these are our various funders to which we're most grateful. Thank you very much for your attention.